believe this is recording. Recording. All right, welcome everyone. So today we're entering module 2, and we're going to talk about gene expression analysis, clustering, and classification. So here we are. Basically, we completed the last module, which was module 1 on genome annotation last week. So we talked about alignment and then hidden Markov models. We looked at some very cool algorithms, dynamic programming, and database search, and how to deal with hidden Markov models. And a lot of the tools that you saw will actually apply very, very broadly, well beyond these lectures, well beyond the course, and well beyond computational biology. Can you see everything? Yeah. Good, beautiful. So today we're going to be talking about expression analysis. On Thursday, we're going to be talking about transcript structure, and then next Tuesday, epigenomics, and then about the 3-dimensional genome. And that's going to be module 2. So basically, in terms of the computational foundations, we're going to be looking at more unsupervised learning and expectation maximization, supervised learning, both generative and descriptive models, how to map strings very rapidly, how to test for significance, how to build splice graphs, and so on and so forth. So the goal for today is to, number 1, introduce what is gene expression analysis, and then talk about different ways of doing clustering and classification for gene expression analysis. So let's start with the biology. So there's 2 main methodologies for figuring out how a particular gene is expressed inside a cell. You can measure expression by basically taking the RNA from the cell, doing a reverse transcription, which is basically instead of having from the DNA going to the transcript, reverse transcription basically means going from the transcript to the RNA, from the RNA to the DNA, from the RNA molecule going to a complementary DNA molecule, or cDNA. And then you can hybridize the resulting mRNA molecules with a DNA array. So you can actually build a microarray where you generate in tens of thousands of spots on your microarray complementary probes that are matching each of the genes that you know about in the genome. And once you have that, you can then see with what strength does each spot light up, which is proportional to the number of RNA molecules and cDNA molecules that are present in the cell. Who's with me on this one? Raise your hands. Awesome, good. So technologically, you basically start with RNA from inside your cell, and then you reverse transcribe it, and then you hybridize it. And then the RNA from inside the cell can come from many places. You could basically lyse the cell and only keep the nuclei and get rid of everything else and just measure nuclear RNA. And there's a lot of RNA inside the nucleus. Or you could somehow measure just the cytoplasmic RNA, or you could only select specific organelles and measure the RNA within those. And again, the diversity of samples that you can put through this is huge. You could basically look at different tissues. You can look at a whole organism. For example, in the case of Drosophila, you can grind up the whole fly, or you could just take the brain. You can micro dissect the brains of the flies and then look at those, and so on and so forth. And most of the time, what we've been looking at is bulk tissue. So you're going to be looking at dissections of different tissues in the human body and then measuring expression there.
And also the screen is being shared. I think it's just recording.
How about now?
Yeah, now we're good.
Okay, awesome. So that's basically this first part. The second part is all about sequencing de novo. So sequencing used to be extremely expensive. It used to be $1 a nucleotide. And for the $3 billion of the 3 billion nucleotides of the human genome, it cost about $3 billion. So it was just an incredibly expensive project to sequence the first human genome. Nowadays, you can sequence an entire genome for a couple $1,000 bucks. So we're talking about 1, 2, 3, 4, 5, 6 orders of magnitude reduction in just as many years, which is just incredible in terms of the huge transformation in technology underlying genomics. And that is reflected in the switch from hybridizing RNA molecules to things that we synthesize to just simply saying, well, let's just resequence it. Sequencing is so cheap nowadays, we can just resequence it. And what you can do then is effectively sequence short reads from the mRNA itself. So instead of taking these RNA molecules and then hybridize them, we're going to take these RNA molecules and simply count them. Now, the advantage of microarray technology is that you can focus on small regions of the genome. You only synthesize what you care about. So if what you care about is 100 genes in your pathway, you don't need to worry about all these other genes in the genome and all the measurements they will lead to, giving you much more bang for your buck, if you wish. The advantage of RNAseq is that they're digital measurements. You're not just looking at hybridization, you're actually counting molecules, and it is completely de novo. You don't need to know of that gene in advance. So you can basically simply sequence out, whatever RNA is out there, and basically come back and say, well, yeah, this one I recognize, I've seen before, or this one I don't recognize. It's a new gene, or it's a new transcript. Does that make sense? Everybody with me on the second part of this? So basically, the microarray technology can basically have very, very many variations. You could basically build one long probe for every gene. You could build many short probes for the gene. You could tile Kmeans across the entire genome. This is how gene expression analysis in the yeast genome used to be done. Basically, you could either hybridize every one of 6,000 genes or tile the whole genome, and then you could discover new genes. And then, variations of this is to just count the reads mapping to every known gene only, or actually reconstruct the transcriptome de novo, which is something we're going to talk about on Thursday. Okay? So once you have your measurements, you can now start asking, what does the data tell me? And that's where things get interesting. Let's see, hide preview panel. That's a good idea. Show the panel. Anyway, I'll figure out how to hide this eventually. So basically, once you have measured, say, 20,000 genes in hundreds of conditions, what you can do is study the resulting matrix in many ways. The first way is to just simply study all of the genes in each experiment. So basically go down the gene vector. And every experiment gives you basically one more column of data of 20,000 genes in that additional condition. And what you can do then is study the expression profile of a gene across conditions. So the beauty of having so many different experiments and all of the genes measured in each of them is that you can actually start building profiles for how the gene is expressed. And once you have that, you can actually start asking questions about how similar are two genes in their expression patterns across different conditions, or how similar are two conditions across all the genes. Okay? So a lot of the questions that you can ask come from the fact that we have hundreds of conditions and tens of thousands of genes, and therefore you can actually start studying the resulting matrix in interesting ways. And the two primary techniques that we're going to talk about today, motivated by expression analysis, is clustering versus classification. So what clustering does is that it basically says, can I find genes that are coordinately active across different conditions? Or can I find tissues in the human body that have similar expression patterns across all their genes? So the goal of clustering is to effectively discover groups of genes or groups of conditions that are similar and that likely come from the same category, and in doing so, reveal some hidden structure about the data. And that's a form of unsupervised learning. So we don't give you any labels, we just say, find patterns in the data, and to do that, you basically just look for similar genes or similar conditions. With supervised learning, you have some known classes. You basically know that, you know, these genes are involved in these processes. And you basically say, can I find additional genes that play roles in proliferation, or additional genes that play roles in, you know, B cell function and so on and so forth. And you can do that by basically building a classifier based on the features of that gene. And these features are usually the expression levels of that gene across different conditions. And then learn how to find additional members of that group. So the goal of classification is to extract features from the data that best assign new elements to one or more predefined classes. Okay? Everybody clear in the difference between clustering and classification? Any questions so far? So again, more abstractly, we have a set of objects. Every object can be a gene or a protein or, you know, a cell and so on and so forth. And they're characterized by one or more features. And usually we're going to be looking at some multidimensional view of the features or a projection of this very high dimensional space. If a gene is measured in hundreds of conditions, then that gene has a 100-dimensional vector, not a 2-dimensional, but 100 dimensions. So you can reduce the dimensionality of that in techniques that we're going to see in the networks part of the class, using, you know, PCA, principal components analysis, and singular variable decomposition. So you can basically reduce the dimensionality of this very, very high dimensional space into just maybe two dimensions or 4, and so on and so forth, and visualize them pairwise. So if you're looking at classifying a tissue, then the vector of features is 20,000 genes in that tissue. Again, a 20,000 dimensional point. So you can basically use that to, you know, classify. So classification is basically supervised learning. You have labels for some of the points, and you want a rule that will accurately assign labels to new points. And the subproblem is how to select the best set of features. And the metric is how well do I classify? And that is a well-defined metric because I know what my answer is. So I can hide the labels for some of the points and basically say, can I classify them well? And that gives me, you know, false positive and false negative rates. For clustering, you have no labels. You simply group the points into clusters based on how proximal they are to each other. And you identify some underlying structure in the data. And the metric then comes from independent features that were not used as part of your clustering to basically say, well, let's go measure a few more things about those genes or about those cells, and then learn something about those, okay? So in every case, we have this, you know, multi-dimensional feature space. We have, you know, genes, proteins, or anything else. And then we classify the points, or we just simply group them into some number of classes without knowing in advance what the classes are. Raise your hands if you're with me so far. Awesome, beautiful. Great. So now let's talk about clustering. So basically we're gonna start with this unsupervised learning where we don't have any labels. There's two types of clustering. One is partitioning, where you basically say, I'm gonna find K categories, and then I'm gonna partition my data into those K categories. So it divides objects into non-overlapping clusters such that every data object is exactly in one subset. And, you know, we're going to see relaxations of that, where you can, you know, have an object belong probabilistically to more than one. The other class of methods is agglomerative. And there, you don't have a single clustering. You don't have a single set of, say, 6 classes. What you have instead is for 10,000 points, you basically have 10,000 clusterings, or, you know, 9,999 clusterings, where basically at every point of your tree, you basically draw a hierarchical nested structure that allows you to sort of say, what are the 2 points that are nearest each other at any one level of that hierarchy, grouping points into progressively larger and larger clusters. And to find a clustering there, you just draw a line across your tree, and you just choose all the different subsets there. So it's a set of nested clusters organized in a hierarchy. Okay? So then that's how we're going to organize the lecture. So first we're going to talk about K-means clustering, so clustering by partitioning. Then we're going to talk about hierarchical clustering, clustering by agglomeration. And then we're going to talk about classification, specifically the example of Naïve Bayes. Okay? So what's the basic idea of K-means clustering? The basic idea is that we assume there's a fixed number K of clusters. So that's given as an input. And you know, you can vary that, and we can talk about ways to select K. And then you partition points into K compact clusters. So our metric is going to be compactness, and we're going to define that. And then the algorithm for K-means is going to be very simple, very similar to what we saw before with unsupervised learning. We basically initialize the cluster centers randomly, and we repeatedly assign points to the nearest center, and then move the centers to the center of gravity of the points that are assigned to each group. Okay? And then we're going to stop when we converge, basically when there are no more reassignments as we go through this. If nothing moves and nothing changes, we're done. Okay? So here's an example. So I have a bunch of red points, and I randomly initialize my clusters this way. Then I assign the data to the nearest clusters. Then I label the data according to the color of those clusters. And then I'm going to recompute the mean of these points that are assigned to that cluster and move the centroid to that. Okay? So now I'm going to move those means and end up with that. Okay? Notice here, these 2 points, even though visually they're closer to the purple cluster, were colored green. Because if you draw a circle around green, you capture those points. Sounds good? So that basically means that I perhaps have an incorrect clustering here. But here's what's exciting. The next time around, when I reassign the points, I just forget the colors that I gave to each point, and I just look at the new centers to recalculate the distances. And now those 2 points that were initially drawn green are now assigned to the purple cluster. And then once I've done that, I've moved all the points, all the centers to where the clusters are. And then, after that, there's no more reassignments. And if there's no more reassignments, there's no more center changes. Okay? Who's 100% with me here? Awesome. Great. Any questions so far? Yes?
So if you have a data point that is equidistant from 2 or more centers, you just randomly choose one and
Correct. So basically your code can be usually deterministic, and it will pick the min, and then it will update the min as it goes through. So basically chances are the first cluster that it needs will be the one that will be assigned. But you could also flip a coin and assign 100% of the mass to one of the 2. But we're going to see a variation of K-means called fuzzy K-means, where you can actually probabilistically assign a point to multiple centers. Other questions? Yeah.
How do you know how many clusters to assign?
So there are methods to find that. You could basically, I mean, what most people do is that they try some reasonable number, and then they see how it looks, and then they try a bunch of numbers. There's a statistical problem there in the sense that you're changing the results by looking at the output, and therefore you should be correcting somehow for the multiple hypothesis testing of the K tries that you've had. There are formal criteria that basically say how well do I capture my data, penalizing for the complexity of my model, and the complexity of my model is proportional to the number of parameters, and therefore to the number of clusters. So there are many ways to do that. There was one more question? Yes, same question. Other questions? All right. It's pretty much a good question at every lecture. So at every lecture, if you just wake up and you're like, what's going on? I'm going to ask something to say, how do I choose the number? And it's always a good question. And the answer is, I truly, frankly, don't have a good answer. But there's many. The problem is that when you have many good answers, it's that there is no single good answer in person. All right. So we're going to be referring to these as the E step and the M step. So in the M step, I'm basically updating the centroids to the mean of the points. And then in the E step, I'm reassigning the points, okay? So now let's look at what are the actual computations that we're doing. So when we're reassigning every point, xi to the nearest center k, what I'm basically doing is minimizing the distance between that point and that centroid. So basically what I'm doing is that I'm minimizing this distance, dik, between point I and centroid k, which is simply, some Euclidean distance of the, 20,000 dimensional vector x, the 20,000 dimensional vector m or mu, and then, the difference between them squared, which is a good measure of distance, basically. Okay. Now, then I update the center mu to the mean of the points that are assigned to it. So basically the next, at iteration n+1, the new vector, 20,000 dimensional, say, vector mu k, is basically the average of all of the coordinates of each point divided by, the number of points that there are. So basically it's just that average summing over all the x's that have that label j. Okay? And then this is simply the cardinality, the number of xi's that have label k. So basically, this is the Euclidean distance, and this is the averaging of those points to basically recompute the centers. Yes?
You normalize the vectors before you start the process?
It's a very good point. So basically, what comes out of your measurements? What comes out of your measurements in the case of RNAseq data is counts of reads. What comes out of your measurements in the case of microarray data is relative ratios. So in the case of counts of reads, there's a problem there, because if I sequence twice as deeply, all the counts are going to be multiplied by 2. So if I have one experiment sequenced at 20 million reads and another experiment sequenced at 50 million reads, they're not going to be exactly comparable. And same for the hybridization. Basically, my array can hybridize longer and so on and so forth. And, that can vary from experiment to experiment. So absolutely, the first thing to do is normalize. So basically what we usually do is that we normalize the data to have 0 mean and standard deviation 1. And then after you've rescaled and renormalized each of the experiments, you then can carry out your analysis. Yeah?
The specific points at which you randomly initialize, is that?
Absolutely, absolutely. So basically, just like we saw before, initialization matters. And what you're doing is actually climbing some kind of optimality space, and you're arriving at a local maximum. You don't know whether it's a global maximum. So what you usually do is that you initialize a few times, and then you see, how did I get to the maximum? What you can also do is randomly sample points to initialize. Instead of just picking centroids in the middle of nowhere, you can just sample points and initialize that way. Okay? Yeah, sorry.
Is this still useful like the RNAseq, for example, the way is this like the columns of things?
So there's something known as the curse of dimensionality, which is that as you add more and more and more dimensions to your data, points that were previously close are now starting to become extremely far apart. So basically, in 2-dimensional space, we're all pretty close to each other. If this was a 20-dimensional space, you'd feel it, realize just how far you are from your classmates. And it's the same thing with points. Basically, as you increase the dimensionality, the data becomes sparser and sparser, and points become further and further from each other. So one way to deal with that is what I mentioned before, so dimensionality reduction. So you're projecting your data into a lower dimensional space that captures a lot of the variation. And that's one way to deal with that. And we're going to talk about that in a couple of lectures, 5 lectures. Any other questions? All right. So what are we optimizing? So basically, Kmeans is an algorithm. It is a recipe. It basically says, break the eggs, then add the flour, add the milk, etc. Sugar or salt, you name it. It's a recipe. It doesn't tell you whether this will be optimal. It only tells you to follow those steps. So will it actually become optimal? So we can actually reason about an optimality criterion for Kmeans. We can basically think of Kmeans as trying to create clusters that minimize some kind of cost criterion associated with the size of the cluster. Before we mentioned we wanted to have compact clusters that capture our data. So what we can do is define some cost function over a set of points, which is basically summing over all of the centroids, all of the clusters, and then summing every point in that cluster, and then minimizing the total distance between points and their respective centroids. Okay, so in order to minimize some kind of cost, to basically create the most compact clusters, we'd like to minimize this function. We'd like to minimize just the distances between blue points and blue center, purple points and purple center, and green points and green center. Okay, I don't care how far the purple points are from the blue points. I only care about, and I don't care about how far the clusters are from each other. All I care about is have I minimized that total distance. Okay, so what's a good way to minimize the total distance for, I don't know, 7 points? How many clusters should I pick? Yes, basically the problem with this metric is that I can always get to zero distance. Just pick as many clusters as there are points and I'm done. Have I learned anything about the data? No. So this is called overfitting, okay? So basically overfitting, you know, in the simplest form is exactly that. It's picking as many clusters as there are centers, there are points. Minimize the distance criterion, learn zero, learn nothing. Why? Because it doesn't make any generalization. You have simply created so many parameters that you can fully capture your data, but it doesn't generalize to a new point. I get a new point in there. What cluster does it belong in? I have no idea. Okay. So instead, for any fixed number k, which is smaller than the number of points you have, or usually much, much smaller, then that optimality criterion actually makes sense. Okay. And then you have to think, you know, if I try a bunch of random initializations, am I going to get to a better, more compact set of clusters? Okay? Yeah. We're going to see various variations of this, but the trouble with mean is that extreme points can make a very big effect. So if I have a bunch of points here and one in outer space, I'm going to think that I'm halfway between here and, you know, Jupiter, and there's nothing happening between here and Jupiter. It's mostly empty. Whereas if you basically think of median, I'm going to end up either on Earth or on Jupiter, but, you know, not in space in between. So yes, absolutely. So median can avoid outliers, or you could run a pre-processing algorithm that gets rid of the outliers first, and then run Kmeans. Any other questions? Yeah.
Is there mechanism for knowing when to add or remove any of these clusters?
Good. So there's two ways to pick the number of clusters. You could just in advance say, I want 7 clusters, and that's it. Or you could basically say, let's try 10 clusters. And after you do your clustering, you could basically say, well, what if I, you know, find that one of the clusters is a little too diffuse? Maybe I want to recluster that. What if I find that 2 different clusters are in fact just on top of each other? Maybe I want to recluster that. Maybe I want to split some clusters, merge other clusters. And I can optimize the number of clusters that way. So that's, you know, absolutely one way to do it. So it's a sort of data-driven approach to the number of clusters. Yeah.
Going on, how do you statistically evaluate that testing?
In terms of paying for the multiple hypotheses? Yeah, so yeah, I mean, of course, any kind of iterative method has this kind of challenge. So yeah, there are ways of dealing with that. There's another question, I think. Okay, good. So here's the cool thing. Kmeans is actually minimizing exactly that function. So what we're going to see is that even though we came up with it as just an intuitive algorithm that made a lot of sense, it is actually optimally solving this particular, you know, optimality criterion, this particular cost criterion. So if I want to basically minimize this total cost, so what I want to do is minimize every cluster term separately. So basically take the derivatives here and basically, you know, this is the optimum. What I want to do is minimize that. So I'm going to expand my square. It's going to be x squared here plus mu k squared here minus 2x mu k. And then this is going to be effectively the sum of x squared, this, this term, which does not depend. So basically this is summing over all the x's, and then the mu doesn't depend on the x's. So this is going to be simply the number of points times mu. And then this part here, again, the mu doesn't depend on the x's. So I can just take the mu out, and it's the sum over all this. And if I'm trying to minimize that, then what I can simply do is basically take the mean of this, which is effectively getting at that solution. So basically, the solution of setting mu k to be exactly that average effectively minimizes that solution. So this term doesn't matter. And then I'm trying to minimize this term here. And then, this term becomes minimal when these two coefficients, match up. And that's effectively giving you exactly the center. Okay? So minimizing this very simple criterion effectively means setting our means to be, exactly that. But the problem is that some of the points can be exactly halfway. So basically, again, this is, K-means as you define it. And that was one of the questions. Some points can be exactly halfway between two centers. So what we could do is probabilistically assign to, basically flip a coin and either assign to one or the other. Or what you could basically do is assign the points partially between them. So you're going to say that this point here belongs to both cluster purple and to cluster green with some probability. Okay? So that's what fuzzy K-means does. Okay? So what fuzzy K-means does is at the when I reassign the points, I'm going to reassign them to all of the centers every time. Oh, sorry. Yes, there's a question.
On this slide,
Yes.
so for the optimality, you showed that you already have a label for each point.
Exactly.
And the way that you're deciding the mean, that as well.
Yes.
But not in terms of actually assigning the labels.
Yes. So we're actually going to, I was surprised that this was not on the slides, but I agree. What you need to do is optimize both terms, basically. And I think there's a slide later on that does that. So it should have been here, but I think there's a slide later on. And basically, when we're going to look at the machine learning formulation of K-means, we're going to see that both the E step and the M step are in fact maximizing exactly that. So you're absolutely right. Basically, assigning the centers to the center of mass is sort of what we showed here, but also assigning the points to the nearest center is the other part of that optimization. Yeah. Any other questions? So basically, again, when we're reassigning every point, we assign it to all of the centers weighted by their distance. So for every point, I calculate the probability of membership for each category, which is simply some probability of being in, label K, given my point coordinates and the centroid coordinates. And then I update the center to the weighted mean of the points assigned to it. So basically, the mean at the next iteration, n plus 1, becomes the the sum of all of the point coordinates weighted by their probability of membership divided by the total number of points, again weighted by the probability. So basically, regular K-means is a special case of fuzzy K-means where that probability is 1 if it's the closest point and 0 otherwise. Yeah.
Again, in the sum, you would be summing over all the points, not just points with the label.
Correct. In this particular case, this probability is going to be very high for the points that are nearest, and therefore I'm going to be weighing much more the points that are nearby, and only letting the points that are far away influence me a little bit. Okay, so it's all here. There's now, oh, I see, not just with label J, but sum over all points X. Yeah, correct. Okay, we'll fix the slides. All right, so that's what fuzzy K-means does, okay? And now let's actually step back a little bit and reinterpret everything that we saw. It was a nice little recipe, but now let's reinterpret it as a generative model. So what Kmeans does is that it effectively assumes that there are these centers that generate points, that spit out points. It's a generative model. And from these, hidden model of the world, I don't really know the true centers, but I generate a bunch of points by sampling from those centers, okay? Basically, there are samples drawn from normal distributions with unit variance, a Gaussian mixture model. And basically, the probability of emitting a particular point X from a particular centroid is, simply following exactly the Gaussian distribution. So basically, the exponent of this times that. So given only samples, how do we estimate the maximum likelihood model parameters for the centroid definitions and for the point assignments? So basically now I have a Gaussian mixture model. I can generate points, or given a bunch of observations, I can basically re-estimate what are the distributions that they came from, okay? So again, the expectation maximization solution, exactly like the Baum-Welch algorithm in hidden Markov models, is that in the expectation step, if the clusters are known, then I can estimate the membership. And then in the M step, if the assignments are known, I can recompute the centroid, okay? So if I know the centers, I can basically estimate what is the probability with which every point belongs to each center. And once I have a set of assignments, probabilistic or not, I can basically calculate the maximum likelihood estimate of what are the parameters of the centroids of these distributions that gave rise to the points that I observed. Okay, so I can choose, the centers, and I can choose the labels that maximize the total probability of observing the data given the model. And then the solution to that is exactly the K-means algorithm. Okay, it's actually the fuzzy K-means algorithm. So what I'm going to do is that I'm going to choose, the set of centers and the set of labels that maximize the probability. And what I'm going to do is that I'm going to basically maximize over the possible center definitions this particular value, which is, the log of the probability of the product of the probabilities of all of the points being generated from the corresponding centers. And then that's nothing more than the maximum of this Gaussian model. And because I'm taking the logs, I can basically now take the sum. And, basically the log of the product is the sum of the logs. And then the log of this is, again, taking the exponent and expanding it out. And this term doesn't matter here, but basically argmaxing this effectively means selecting the points that minimize this Euclidean distance, okay? And then the solution to this is effectively the centroid. So the EM solution of this, selecting both the centroid definitions and the assignment of points to centroids, maximizing that total probability of generating my data from the underlying model parameter is effectively equivalent to minimizing the centroid distance. Okay? Who's with me so far? Raise your hands. Awesome. Good. Who thinks this was kind of cool? We started with this recipe, seemed like a good idea, and then we started seeing how it optimizes these geometric criteria. And then we reformulated the whole thing in sort of this generative model space, where you have some hidden model of the world, which is generating data. And then we've used Bayes' rule to now ask, given that we've observed a bunch of data, what are the most likely centers that generated these data? Flipping the directionality of from generative to inference, and then ending up with exactly the same solution. So this is basically how do we choose the center definitions, and then how do we choose the assignments. So what we want to do is for every point X, I want to choose a label K that maximizes that total likelihood of producing point X from the center. And then again, that's maximizing this, which is the same as minimizing that distance, given the minus here. And then that means that the solution over all possible clusters K for what label I want to assign to my point X is effectively the nearest center again. So that's sort of the second half of the equation that you pointed out earlier. So basically maximizing over all label assignments given the EM solution is, choosing the near center, and then the maximum likelihood estimate is again the near center. Yes?
So in this formulation, and also in the tables, we can only fit. So the cluster shape that is this variable, but I mean, have like cluster shape.
So I believe that's the next slide. So it's a very good question. So basically, I'm going to come back to that. But basically, your question is, wait a minute, you assume that in this two-dimensional space, however you drew that space, that the variation in this dimension and that dimension are, number one, uncoupled, number two, the same. That basically the covariance is, 0, and that each of the variance in unit variance along each of the dimensions. We're going to very soon see how we can overcome these assumptions. Yeah.
Can you do something similar with expectation maximization to also have the number K automatically generated?
You could iterate over a bunch of Ks and then evaluate the score for each of those, penalizing for model complexity. And that penalty is where the art is at in terms of choosing what is my penalty and how does it depend on my number of parameters. Other questions? Okay.
Just regularization.
Yeah, pretty much, but you can choose different types of regularization, basically. All right, so here's one of those tables that you're going to see across many lectures. The goal is to sort of, look at step back from the lecture and look at the various formulations. So basically we saw for Kmeans an algorithmic formulation and a probabilistic interpretation. And then for fuzzy K-means, again, an algorithmic formulation and a probabilistic interpretation. The algorithmic formulation was basically first initialize the set of clusters K and the centers, assign every label to the nearest center, move the means to the center of these points, and then iterate. And the probabilistic interpretation was to again initialize the model parameters, estimate the most likely missing label from the previous assignments, and then choose the new maximum likelihood parameters given the points in the label. And here, we're choosing exactly one label. With fuzzy K-means, it was exactly the same thing, but instead of choosing only the nearest center, we calculated probability of membership. And then instead of choosing the centroid of the points, we chose the weighted centroid, weighted by the probability. And then here, instead of estimating the single most likely missing label, here we estimate a probability over missing labels, given the previous parameters. And then we choose the new parameters to maximize the expected likelihood given the label estimates instead of just choosing the single max likelihood. Okay? So basically this is weighing over all of the points that are assigned. Okay? And then the cool thing with the expectation maximization algorithm is that there are actually some theoretical guarantees. You're actually guaranteed to increase the likelihood of observing the data from your model at every iteration. You're not guaranteed to achieve a global maximum, but you're guaranteed to converge to a local maximum. Now, we can now go back to your question and say that, hey, EM, now that we have this more complex probabilistic model, is actually much more general than fuzzy K-means. In particular, the K-means solution assumed uniform priors, basically that every cluster had the same size. I was equally likely to sample points from the red cluster as the blue cluster, but sometimes there's another cluster that has many more points. So basically there, the prior is no longer uniform. You can actually have some class-conditional priors, and you have some probability of the class, which basically tells you that some classes have simply more points. So if I sample at random, I'm more likely to find, a gene involved in metabolism than a gene involved in I don't know, the transport of some molecule, okay? The spread of points, we had assumed previously with Kmeans, we could only deal with unit distance functions, whereas here we actually have a Gaussian distribution where the sigma can actually be tuned, not just the means, but also the standard deviation. We had previously with Kmeans only allowed symmetric clusters where the different dimensions were independent of each other. Whereas here we could actually have some covariance where there's some relationship between X and Y, and you can express that this way. And for the label assignment, with K-means, we had fixed only the max. With fuzzy K-means, we were using the full density. And with EM, we could use, again, the full density, or we could simply sample from that posterior. So instead of always choosing the maximum, I could basically say, well, I'm halfway between these points. I'm going to basically flip a coin according to the probability of membership. And then I'm going to put my entire weight on the label that came up on top. So if I have a point here, this green label will have much, much higher chance of being the true assignment, but this will have some smaller chance. And every now and then I'm going to assign it entirely 100% to the blue point. And that's what Gibbs sampling does. So EM is using the full density to basically assign the point probabilistically, weighing all of the clusters and all of the points and all of the evidence. Whereas Gibbs sampling effectively looks at that probability and then samples from that probability. Okay? So basically, with the original data, if the original data was this, then the K-mean solution would basically assume that every cluster has the same number of points and unit variance, and therefore would assign the points this way. Would basically say, well, hey, there's one cluster, there's the other cluster, there's the third cluster. And that's all K-means can do. But if I have a full EM model, allowing for different priors, for covariances, and different Gaussian distributions, then I can basically say that this will end up with more prior, a stronger prior, so more points, and the covariances will be as such. Yes.
I thought you mentioned if we figure out the distribution for the EM is the same as the fuzzy mean.
If you assume that all of the class priors are identical, that all of the Gaussians have unit variance, that the covariance is 0, and you use the full density. So basically, the first formulation of fuzzy K-means was in fact it was a special case of the EM generalization. Yeah.
So when you say that class priors is the non-standard field, you're saying like we're saying something about the clusters before we start the algorithm?
No, what you're saying is that you're allowing the algorithm to adjust by saying, oh, I find a lot of. So instead of forcing a solution where all of the clusters have the same number of points assigned to them, it's going to say, well, it seems that this cluster has more points. I'm going to increase its prior.
Oh, so that's
Exactly. It's one more parameter that you can tune. So the prior becomes an explicit parameter rather than always being set to identical values across all of the points. It becomes a tunable parameter that you can tune as you go. So you're simply allowing the model enough complexity to vary the prior. You're not saying in advance, here's the higher prior. Does that make sense?
Distribution per cluster as well. And that's also like the new higher also. Parameters be trained.
Correct. But that was the case for all of the formulations. Basically, the mu I were these hidden parameters, and we were adjusting them. But we had assumed a uniform prior without talking about priors. We had basically, it's just that very simple implementation of the Kmeans algorithm was in fact optimizing a unit variance and, uninformative priors. Taylor, it seems like there's something wrong with the meeting. Sorry, what were you saying? Sorry.
So your data dimensionality is 20,000.
Your data,
Let's
what?
assume that your data space is 20,000 dimensional.
Yeah, sure.
Then your covariance matrix will be 20,000 by 20,000.
That's a lot of parameters.
Yeah.
Yes,
So
yes.
that will be
Yeah. So with every increased you know, parameter that we have, there's a model complexity that we have to pay for. And chances are that we just simply won't have enough points to train such a big model. So that's why we probably won't allow that. Yeah, correct. Exactly. Dimensionality reduction is definitely the way to start. Yeah. Other questions or comments?
You need to share your screen.
Okay. Okay, do you see it?
Yeah.
Great. Cool. So what have we seen? We've seen expression clustering and we've seen HMMs. And we saw some parallels between the two. And in a couple of lectures, we're going to see motif discovery. And then we're going to see some parallels again. Okay? So let's now make those parallels explicit. So with Kmeans, we assigned every point to the nearest cluster. And that was simply picking the best. And in the HMM space, the equivalent was basically Viterbi training, where we were labeling the sequence with the best path. Okay? With expression clustering, we had hidden labels, which were the cluster assignments, and also the properties of those clusters. And then similarly, with HMM, the hidden labels were the state path. Basically, what are the hidden labels of every character in my sequence? And then the the parameters of that path were basically the emission probability and the transition probabilities between those. Okay. So here we have K-means, which is equivalent to Viterbi training. And then fuzzy K-means was assigning every point to all the labels probabilistically. And then that was equivalent to the Baum-Welch training in HMM space, where you label the sequence with all of the paths, which was posterior decoded. Here I alluded to this Gibbs sampling approach, where you basically, instead of assigning each point to all labels probabilistically, you're going to pick one label, but you're going to pick it at random based on their relative probability. And that would be equivalent to sampling a single label for each position according to the posterior probability, or assigning each point to a random cluster, again, by sampling by their proximity, and so on and so forth. And we're going to see this much more formally in the motif discovery space with Gibbs sampling. Okay? But all of that falls within the same family. And then, of course, once we had that, we updated the model parameters by either averaging those points assigned to the label or of averaging of all points weighted by their membership. And in this particular case, we're just going to be averaging the points that were assigned to the label, which was some kind of sample. Okay? All right, so that was K-means, basically clustering by partitioning. We saw first the algorithmic formulation, the update rule, optimality criterion, and then fuzzy K-means. And then we saw the machine learning formulation with a generative model, sampling from a mixture of Gaussians, and then expectation maximization for the solution. Yeah.
So I think this point was touched on before the lecture as well. But so Kmeans does clustering entirely based on Euclidean distance between labels. And now when we are operating in such huge dimensions, won't the noise be a very big concern? Because
noise in your measurements, basically.
Noise in measurements, because now you have like noise across 20 features that add up and actually, because the number of features is so high,
Yeah.
And trump over the signal and lead to the points being misclassified, given that like entirely the
So, what are some recommendations? What are some ideas of what you could do? Again, so the same thing applies over and over again. So basically, what dimensionality reduction allows you to do is basically say, if I were to choose a lower dimensional space where my points are coming from, then I could effectively reproject that lower dimensional space back onto the higher dimensional space and look for deviations between the original measurements and my lower dimensional projection onto the same values. And then say, well, these points seem to be way off from what I would expect if the data was truly coming from a lower dimensional space, where, for example, there's only so many gene regulatory programs in the cell. And therefore, the expression of those genes is constrained. Not every gene can change completely at random. They change according to sort of the regulatory circuitry that controls these genes. And since that regulatory circuitry is not infinitely rich in all of the regulatory signals that it can use, you can basically say that a lower dimensional representation is probably more appropriate for that. Does that answer your question? I mean, it's a very, very good question, but yeah, I mean, that's sort of one way of dealing with noise. There's other ways. You could do replicate experiments. You could do imputation. You could basically say, if I look at all of my data, are there some patterns that I can use to predict the expression of every gene from all of the other genes? And when I do that and I find discrepancies, does that indicate that perhaps the measurement of that gene is inaccurate, that there's something wrong with my probe, and so forth? Other questions? Okay. All right, so now for something completely different, we're going to look at hierarchical clustering. So now, first of all, take a stretch break. So hierarchical clustering is completely foregoing the question of how many clusters are there. It basically says, you know, let's just ignore that. So it's basically, a lot of people asked about K. We can always make the clusters more compact by increasing the number of clusters. What happens if K equals the number of data points? You know, great score, but not meaningful at all. Are you still on?
No.
So what hierarchical clustering does is that it basically says, let's just ignore that. Let's simply start with every point being its own cluster. And at every point, at every step, choose a pair of clusters, initially points, and then groups of points, and then merge them. So basically, if I have these points here, merge them, and then do I need to reshare?
No, we're good.
You see with the slides?
Yeah.
Awesome. You still see them?
Yeah.
Good. So basically, I merge. Okay. So what I end up with is first merging D and E, then merging F with D and E, merging A and B, merging D, E, F with A, B, merging C with A, B, C, D, A, B, D, E, F, and then merging G and H together, and then merging the whole thing. So here's the series of merges that I did when carrying out this process. So merging those, and so on and so forth. So this is exactly the same algorithm that we're going to see in the phylogenetic lecture, and this is known as UPGMA, or unweighted pair group method with arithmetic mean, which is not a super creative name, but it basically says that I'm not going to be weighing the points, I'm only grouping pair at a time, and then arithmetic mean is sort of how I compute the next set of centroids. Okay, so how do I choose a number of clusters? What I can basically do is simply draw a line here along the hierarchy, and wherever the tree gets cut, this is the underlying clusters that I have. Okay, so A, B, D, E, F, C, G, and H. Okay, and if I want 3 clusters, I just move one up and I just cut here. Okay, if I want 4 clusters, I just move one down, cut here. Okay, it's that simple. Of course, there's a lot of stuff hidden under the rug here. So first of all, we sort of assumed that we were going to recompute the distances between these two and that guy. The way to do that is to basically, compute a cluster-to-cluster distance based on a point-to-point distance. And how do I do that? Well, I can either take the closest set of points and say that my cluster distance is the distance between the closest pair of points, each belonging in a different cluster, or, and that's called single link, or complete link basically says that, to bring these two clusters together, I'm going to have to worry about how far apart are the furthest points that are going to belong to the ultimately merged cluster. Or average link could basically say what is the full set of pairwise distances between all points here and all points there. So, 3 times 2. And then take the average of those. Or I could simply average the points in this cluster, and then compute the distance between those two averages. Okay? And then again, the cluster distance metric affects both the results and the runtime. In this particular case, the runtime is much faster because all I need to do is sort of choose the distance between one point. But then the problem is that to find that distance between those 2 points, I have to search through all the points. Or I can use some interesting data structure to basically remember what points are near each other in the space, and then only calculate distances there. Here, again, I have to compute the n squared set of all distances and then average. Here, this is actually faster at the level of clusters, but it basically means that every single time I add a point to a cluster, I have to recompute its average and recompute the distance between that average and all the other points. Okay? So there are, again, advantages and disadvantages for every one of these metrics computationally. And also biologically, it sort of really matters. Okay? Yes?
Could the single line method be done with natural logarithm?
I believe so, but let's talk after class. Yeah, I have to chat about that. Okay. Any questions on this? And again, these are the cluster-to-cluster distances. But then how do I compute the distance between these 2 points? Even that is subject to various choices. So I could choose Manhattan distance by basically saying, you know, I can only walk along city blocks. Or I can use Euclidean distance. Or I can use, you know, more complex distances. I can use correlation. If all that matters is not the absolute points, but how, you know, the deltas, then correlation will basically be fine. I could use rank correlation, which can be more robust to outliers instead of sort of, you know, being very influenced by points in outer space. I just simply say this is just the furthest point. Or uncentered correlation, you know, where I'm basically calculating some kind of angular separation, or you know, other various metrics. Okay, so then the cluster-to-cluster distance is again a function of this point-to-point distance, again giving you very many choices. And then of course the problem is that once you have your clustering, you'd like to evaluate the results of that clustering. So with Kmeans, we were optimizing, turns out, some criterion of this compactness of the clusters. How do I evaluate that criterion for hierarchical clustering? And again, this approach also applies to partitioning clustering. So to evaluate the cluster performance, you know, we'd like to know some kind of robustness. We'd like to, for example, randomly select samples from your data set and then cluster those and then repeat. And if you have truly robust clusters, they will show up in all samplings. So if you keep sampling subsets of points, the same clusters will emerge. And if they don't, you should basically worry about whether this was just an example of overfitting. Or alternatively, you could basically say, look for categories of genes or of cells or of tissues that are overrepresented in your clusters. And this is something that's also used in motif discovery. So basically, how do you evaluate the clusters? What you can use is your hypergeometric distribution. You can basically select k elements at random and say, what is the probability with which those k elements will end up in the same color? So if I choose a cluster size k and I find, say, p points that are labeled orange within that sample, I can say, well, could I have obtained that just by chance? And one way to do that is something known as the hypergeometric distribution, which is to calculate the probability that the number of positives, say, orange, is greater than whatever value I found here, which is simply summing over all possible, number of such points, and basically saying what is the chance of, out of m points choosing p, and out of k minus m points choosing n minus p, out of p points choosing m, and out of n minus p points choosing k minus m, that respectively belong in these clusters, out of all of the possible ways that I could choose k points out of n. Okay? So that is the probability that a randomly chosen set of k experiments would result in m positives and then k minus m negatives. So basically I have n experiments, I cluster k elements, and then what I want to calculate is the p-value of a single cluster containing k elements, of which at least, r are positive. And basically you can, just simply count the combinatorics of all of the ways of selecting that. And this is effectively what is used very often basically saying, great, I chose a set of k elements. Let's now see if they are functioning in a common pathway. And I can use a hypergeometric distribution to basically calculate that. So you can evaluate those based on all kinds of functional enrichments, based on motif content, as we're going to see in a couple of lectures, and so on and so forth. Okay, so that's basically clustering, either using Kmeans or hierarchical clustering, either by partitioning into a set of k clusters or by agglomeration hierarchically all the way up. Now let's turn to classification. That's basically when we have, yeah, questions? Yeah. That's basically when we have labels, okay? All of this was unsupervised clustering, but supervised clustering, unsupervised learning, but supervised learning in many ways is simpler. So what we're gonna see is that Naïve Bayes has in fact a lot of the same underlying infrastructure that we saw in the machine learning formulation of Kmeans. Okay, so this will not be very foreign. So again, there's two approaches to classification. You can have generative classification, where you basically build, build a model of the world with some features and some distribution of those features in a class-conditional fashion. And then you can generate data from your model, or you can say what is the most likely parameter, set of parameters that could have generated the data that I'm observing. So you use probability calculus for making these decisions. Discriminative classification, which is support vector machines, for example, that we're going to touch upon briefly, has no modeling of the underlying distributions, and it simply makes decisions using a set of distances from a boundary. And then, this is equivalent to what we saw for a generative model of hidden Markov models versus a discriminative model for conditional random field. So Bayesian classification with a single feature can be boiled down to this. What you have is an underlying distribution of red points and blue points. And then you sample points from those distributions. And when you end up with something all the way out here, the red distribution is much more likely to have generated. When you sample something down here, the blue distribution is much more likely to have generated. And then in between them, there's some kind of, uncertainty region where you can't really classify things. And then you can choose a cutoff for choosing a point to be in that class according to whether it is, above some particular threshold. And basically, regardless of what these distributions look like, in the end, you're going to be making a decision based on a comparison of a value that you're observing and then some kind of threshold. And if it's above that threshold, you're going to say it's a red point, and if it's below that threshold, you're going to say it's a blue point. So classification really boils down to that. How do I choose that cutoff above which I'm going to say blue and below which I'm going to say red? And of course, I'm going to be doing this in a very high dimensional space. This is just taking one of those dimensions, and then you can do the same thing across all the other dimensions. Okay? So if you know the distributions, then how do you classify a new example? You can basically pick a cutoff, and then what you could do is minimize the classification error. That will give you some cutoff here, or you might maximize the posterior probability. Or if you have many classified examples already, if you have a very high cost of making a mistake and saying that something is blue when it's red, then I'm going to weigh these distributions according to that penalty function, and that will effectively translate into simply shifting that threshold. Basically, if it's very costly to say blue by mistake, then I'm going to shift that threshold to the left. I'm going to say blue less often when I'm really, really sure it's blue. And basically, each of those, if these are positives and these are negatives, has some false positive and false negative rate associated with it. Okay? So for example, you could say what are DNA repair genes, based on their higher expression during stress. Or you could say that protein coding regions have higher conservation levels, or regulatory regions have higher GC content. In general, you basically have some kind of foreground in red, some kind of background in blue, and you're trying to decide whether something is red or blue. So if you have many classified examples already, you can estimate these model parameters, and you can use parametric or non-parametric models, class-conditional distributions, priors according to this. So basically, if you have many more red points, or if the cost of making a misclassification is different, so you can basically include priors and penalty functions. And then what you always have is some kind of probability of emitting a particular feature given the class, some prior for each of the classes, and then you're trying to use that to calculate the probability that a particular element comes from a class given its features. Okay, so the likelihood is basically what your generative models are about. The posterior is what you're trying to calculate. The prior is what you estimate based on the density of each of the points in each of the classes. And then the evidence doesn't really feature in that classification because you always divide by the same amount. And that's simply, the total P of X summed over all paths that we saw earlier, okay? So basically what you're trying to estimate is the probability of a class generating a set of features given your underlying model of generating a bunch of features from each class. Okay? Who's with me so far? Raise your hands. Awesome. So regardless of what the situation is, you always want to estimate distributions from those and then use that to make decisions. So you want to select a class that maximizes that posterior probability, given this. And the maximum a posteriori estimate is the best class being the argmax over all set of classes of this, which is the same as the argmax over all classes of that, which is basically including the prior and the likelihood. And then the evidence doesn't matter because argmaxing over C is invisible to that, okay? So scaling the above distributions based on the class priors. So you basically have a set of features for each class that are drawn from the conditional probability distributions. So here's the underlying model that we never observed. What we observe are the observations, the green and the red points that are sampled from these classes. And then we can model the class conditional probability distribution using our observations. So what we're going to say is, what does this distribution of features given class 1 and this distribution of features given class 2 look like given the points that I've already observed? And of course, this might be a weird sample. We might have gotten unlucky or you name it, but does that make sense? So now this is only using the likelihood. Now, if we also have class priors, we can basically say what is the probability of having a mitochondrial protein. There's like 1,500 mitochondrial genes out of 21,000. Then the prior being in the mitochondrial class is going to be smaller than being in the non-mitochondrial class. And then the total evidence is, P of feature, which is simply summing over all classes this P of feature given class times P of class. Okay? And it does not need to be known for classification because, it features in all of them equally. So then we can just ignore this and just focus on that. And what we can do is effectively divide each of those by, P of X and simply have this, which is the probability of observing the feature given the class times the prior of each of the classes. So what we can do now is classify as class 1 whenever this is greater than that, and then classify as class 2 otherwise. So instead of having to compute two numbers each time, what I can simply compute is a single number that basically says, what is the ratio of those two? And if that ratio is greater than 1, then I classify as class 1. And if the ratio is lower than 1, I classify as class 2. Or I can take the log of that, and if that log is greater than 0, I classify as class 1, and if it's less than 0, I classify as class 2. Sounds good? So that basically gives us a discriminant function. So it's a single function g of x that I can compute for my data, and if that function is positive, I classify as class 1 and class 2 otherwise. Okay? So now I can basically train my model using a labeled test set, and then I will classify each object in the test set and then count the number of each type. So, effectively doing that, what I can do is either use parametric model that basically says, here's my mean and here's my distribution, my mean and standard deviation. Or I could say, here's a non-parametric model that simply bins the data and then measures just how many elements are in each bin. And every now and then I'm going to get some zeros. And that's where I'm going to deal with pseudo-counting. As we talked about earlier, the problem is that the higher the dimension, the more bins there are going to be. So in a one-dimensional space, I may have 5 bins, but in a 20,000 dimensional space, if I only have 5 bins in each dimension, I have a huge, huge number of possible bins to fill in. And that's a very simple intuition for why I need many, many more data points as I have many more dimensions. Because you're going to have a lot of these zeros all over your space. Very, very few of these entries are going to have any points at all. So to estimate your priors, you can use that using either expert knowledge or use some uninformative priors, or estimate the relative membership of each class during training. You can combine multiple features by simply multiplying things out across each of the dimensions, and then multiplying your discriminant function. And then you can evaluate your classification using the number of true positives, false positives, true negatives, and so forth. And then this table is very helpful. Basically, your true positives are the ones that are truly classified as the class and called that class. And then false positives are the ones that you're calling true, but in fact are false, and so forth. And then there are some examples of how this is done in practice. Basically, classifying a new protein using exactly this Naïve Bayes approach. Okay. So unfortunately, we're not going to talk about supervised machines, except to say that instead of having a generative model that tries to model each of the distributions, what SVMs are trying to do is simply find a threshold that distinguishes classes from each other, and then you can